we can now compare those sequences letter by letter to other species, especially other species that we thought were related to us based on other criteria like embryology and the fossil record and morphology and that sort of thing. We find out that our closest living relative, which happens to be the chimpanzee, that our two genomes are over 95% identical to one another, base pair DNA letter for DNA letter when compared side by side. We also have those same genes in the same order. Not only do we have the same library with the same books, we have all the same books in almost all the same order. There's some changes because things can move around a bit. So another way to put this is that our two genomes are exactly what you would predict as slightly modified versions of an ancestral genome. Here's an image. This is data from the 1980s. On the left-hand side, you see a human sequence, a human chromosome, and then next to it is the comparable sequence in chimpanzees. And you can see many times over, we see the same banding pattern. Here's a region here that's flipped relative, so this bit here is flipped over relative to what's here. But then you can see by the time we're back down here, it's all matching up. Mutations can happen to a gene that remove its function, but leave large portions of the gene's structure intact. So we have confidence that we're looking at the remnants of a gene. When we look at the human genome and compare it with the chimpanzee genome, the gorilla genome, and so on, what we notice is that not only do we have mutations in many of the same genes, we also have exactly identical mutations in those different genes. So not only do humans and chimps, are they mutated in the same genes, but it's the exact same, right down to the DNA letter. Now, the two hypotheses for that would be, perhaps independently in two separate lineages, the exact same mutation happened in those two populations. That's one hypothesis. The second hypothesis is that this mutation happened in a common ancestral population, which was then inherited as those two populations went their separate ways. And that, of course, is much more likely. If you take the data set from this particular paper, this is what you see. We see some mutations that are unique to a specific lineage. So here's 15 that are unique to humans. Here are 3 that are identical between humans and chimps only. Here's another 3 that are identical between humans, chimps, and gorillas. Here's another 6 that are identical between humans, chimps, gorillas, and orangutans. Notice also what we're not seeing. What we're not seeing is, say, a mutation that's present in orangutan and present in human, but absent from chimps and gorillas. If a mutation is present in orangutan and present in human, we can predict with great confidence that that exact same mutation will also be present in chimpanzees and gorillas, because the prior ancestral population of humans and orangutans is also the prior ancestral population of gorillas and chimpanzees. Here's another example that I also like to use, and I talk about this in the book as well. But evolution strongly predicts that mammals that mammalian life is an innovation within a group of organisms that laid eggs. So the idea is that mammals are highly modified egg-laying organisms. The lines of evidence we have is that placental mammals, including humans, share a common ancestral population with amniotic or egg-laying organisms like birds approximately 310 million years ago. So what this group was interested in was simply asking the question, are there the remains of egg yolk production genes in placental mammal genomes like the human genome? What they did was they said, okay, in the chicken genome, here's this vitellogenin. There's 3 in the chicken genome, vitellogenin 1, 2, and 3. They simply noticed, or they recorded, what other genes are present nearby, and then they simply looked for those genes in the human genome. So here's the gene. This is a functional gene that's unrelated to egg yolk production. Similarly, these 2 genes are not related to egg yolk production. These genes are present in the human genome, these 3. And then they simply sequence this area to look for remnants of these genes devoted to egg yolk production. Wherever you see a little black bar, that's a sequence match between the human genome and the chicken genome. So yes, humans have small fragmentary remains of these sequences that are functional egg yolk producing genes in chickens. Those same mutations that we see in the human genome, we also see them shared in some of them in every other placental mammal. So this is something that was lost a long, long time ago. So, like I said before, if we hadn't ever thought up evolution prior to genome sequencing would have definitely brought that idea to the fore.